When sequences are only very distantly related, alignment becomes more challenging. In the case of hemagglutinin and neuraminidase, when we compare the different subtypes, they are much more distant than sequences within a single subtype. That will mean that there will be different lengths, and they will have some regions that vary completely between one another. We actually had to edit the sequence before alignment with muscle, in order to get a good alignment. We aligned the neuraminidase sequences with both clustal omega and muscle. You can see from opening the file in Mega that there aren't many nucleotides that are completely conserved in all of the sequences. This first file was the alignment with muscle. There are quite a lot of gaps, and some of them are a single nucleotide, which indicates a poor alignment. However, we need to look at the clustal omega alignment as well, for comparison. By eye, the clustal omega alignment looks considerably worse with a lot more gaps, although there are also possibly more conserved nucleotides. The next step is to carry out a phylogenetic analysis for both of the alignments. We need to create phylogenetic trees, using the same methods and the same settings, for both sequence alignments. The maximum likelihood tree is the best method and the most reliable. The bootstrap value is set to 50, which is enough. It is a nucleotide substitution type, using the Tomua-Nye model, which is the default. The gamma distribution is set, along with invariant sites, and there are 5 gamma categories. Once you have the bootstrap consensus tree, you need to export the tree in Newick format to a file. Don't forget to export the bootstrap values. It will first give you a window with the text file, which you need to save as a tree file. You can now do the same with the muscle-aligned sequences, making sure not to change any of the parameters for generating the tree, as it should be a fair test. Once you have saved the tree file in Newick format, the only thing left is to compare the two trees. You can do this in Mega or using Figtree. The key things to look for are which subtypes are neighbors and which subtypes form clusters.